Bismillahirrahmanirrahim. Welcome, my dear students, to Senior 1 to a new biology session. Today, inshallah, we'll start Unit 2, Chapter 1, talking about the cell theory. In Unit 2, we are talking in this unit about the cell and the cell ultrastructure, the cell structure and function. Unit 2 is talking about the cell structure and function. We have 3 lessons in this unit or chapters. Chapter 1, the cell theory. We'll finish it today, inshallah. Chapter 2, the cell structure. And Chapter 3, differentiation of cells and diversity of plants and animal tissues. Let's start with the first chapter. We're talking here about the cell theory, the discovery of the cell, and the cell theory. There is many scientists help in the cell discovery. How did they discover the cell? Of course, by using microscopes. Before the year of 1665, before this year, we don't know a lot about the cell, but Robert Hooke, the English scientist, started making a simple microscope in the year of 1665, and he started studying the pieces of cork, the cells in the cork, using this microscope, and he concluded that it consists of a number of tiny vacuoles or holes. These vacuoles or holes, called them cells. He called them cells. The meaning of the word cell from Latin language cellula comes from Latin language or Latin word cellula, which means vacuole. The word cell means vacuole, or a hole, or a small room. So the name cell comes by Robert Hooke. We can say that he is the founder of the cell, and the one who gave the cell this name. After that, by about 10 years, another scientist called Van Leeuwenhoek from Deutschland, Netherlands. In the year of 1674, he made a stronger microscope. And this microscope has a magnification power 200 times. So he can magnify better than Robert Hooke. So Van Leeuwenhoek start watching or examining the living organisms in a drop of water, a drop of pond water. And he is the first one who in the world see the world of the microorganisms. The world of microorganisms, the first one who saw was Van Leeuwenhoek. The first one to see the world of living organisms or the microorganisms like amoeba, paramecium, and so on. He was the first one who saw these living organisms, Van Leeuwenhoek. After that, in the year of 1838, another scientist called Matthias Schleiden discovered that, said that all plants made of cells. All plants made of cells. Matthias Schleiden said that all plants made of cells. Another scientist, Theodor Schwann, in the year of 1839, say that also animals made up of cells. Schleiden say plant made of cells, and Schwann also say animals also made up of cells. Finally, Robert Virchow, he made the theory. He made the theory and say that the cell is a building unit of living organisms and also the function unit of all living organisms, although they are unicellular or multicellular organisms. And he said also the cells are produced only from cells which existed before. Any existing cells now comes from another cell was existed before, of course by division or by reproduction. And finally, the cell theory has three basics. The cell theory has three basics. We have to remember. Basic number one, all living organisms are composed of one or more cells. The unicellular organism body consists of one cell only, and the multicellular organisms, their body is made up of many organisms. And basic number two, the cell is a basic unit and function unit also of all living organisms. The cell not only the building unit, but also the function unit. The meaning of function unit, all biological functions occur in the cell. Respiration, reproduction, growth, excretion, and so on. These processes, the biological processes for life, happen in the cell, occur in the cell. And finally, basic number three, all cells come from pre-existing living cells. This is the basics of the cell theory. You have to remember very well. Basic number one, all living organisms composed of cells or more. The cell is a basic structure and function unit of all living organisms. All the cells come from pre-existing cells. This was the cell theory made by version. Development of the microscopes. Development of microscopes. It was the simple microscopes only used until year 1950. 1950, the scientists start making a very strong microscope called electronic microscope. Before that, only we can watch the living organisms by the light microscope. The light microscope which you can see at school. The light microscope, the microscope that you can see at your school, and you can buy one from the internet and you can study some living organism using it. But in the year of 1950, the scientists discovered how to make the electronic microscope, which is a very strong microscope. We'll study it after studying the light microscope. So the light microscope, as you see, available for scientists until 1950. After 1950, they start making the electronic microscope. It depends on the light. The natural light of the sun or the artificial light, in watching the slide, on watching the sample of the living organisms and so on. So it depends on the light. It can magnify things by cutting them into thin slides, which allow the light to transmit through them. You can make a slice, a small slice, a very thin slice, and examine it by the microscope. If you see here the shape of the this microscope, all the microscope of Robert Hooke and Leeuwenhoek, and this is the light microscope which we are talking about. The light microscope which we are talking about is here in this picture. We can see the structure of the light microscope. Here we have ocular lens. Ocular lens is a lens that you look at, the lens that you look at by your eyes. And we have objective lens, the lens which you put the object which you see under it, the objective lens. And we have the stand or the stage, and we have a mirror here to collect the light and make the light fall on the slides or fall on the sample which you study. This is the light microscope we are talking about. So the light microscope can magnify things up to 1,500 times. The magnification power cannot be more 1,500 times. Only 1,500 times. If you magnify more than this number, the photo or the picture will be not clear. The photo or the picture we cannot clear if you magnify more than 1,500 times. We cannot magnify more than 1,500 times because this makes the photo unclear. And we have two lenses, so we have two magnification powers. We have the magnification power of the eye piece or the eye lens or ocular lens, and the magnification power of the objective lens. If we multiply them, we get the total magnification of the microscope. So if I ask you that there is a microscope with the magnification power of the ocular lens 20 times, 20x, and another lens, the objective lens, magnification power is 10x. So what is the magnification power of the microscope? If the ocular lens magnify power is 20x and the objective lens 10x, so the total magnification will be what of the microscope? We multiply, we multiply 10 by 20. It will be what? 10 by 20 equal what? 200. 10 by 20 equal 200x. So the magnification power of the microscope is the multiplication of the magnification power of the ocular lens and the objective lens. The ocular lens and the objective lens. The total magnification of the microscope is the multiplication of two lenses, the magnification power of the two lenses. But if we have magnification power 40, for example, x, and the objective lens also 40x, does the photo or the picture become clear or unclear? What is the magnification power? Total magnification power here is what? 40 by 40 equal what?
1,600.
Yes. So does the photo here more clear or unclear?
Unclear.
Unclear. Why? Of course unclear because it is more than what? More than 1,500. More than 1,500. So the photo will be unclear because you cannot magnify more than 1,500. Okay? If we have a photo magnified to 100 and another photo magnification 800, which photo is more details? You can see more details, 200 or 800? In which you can see more details?
800.
800. Top. In which you can see more cells, more number of cells, 200 or 800?
800.
No, you can see more cells by smaller magnification, not by bigger magnification. You remember, you can see, yeah, if you see small cells like this, small cells like this, big number of cells, by magnification 200. This is magnification, for example, 200. How many cells you can see now? It is مثلا, for example, 12 or 15 cells. Type. This is 200. If you make magnification 800, you will see small number but more details. You will see smaller number but in more details. Is it okay? 200 you can see big number of cells. 800 you can see more details but less number of cells. Is it okay? You have to remember, by bigger magnification you can see less number of cells but more details. Now, how to use the light microscope. Light microscope, as you see here, we have many parts. This is the ocular lens. This is the objective lens. We have many objective lens. Why we have many objective lens? Because we can use according to the size of the sample which we study. We have many objective lens because the sample which you use, which you study, may be different in size. So you can change the objective lens to change the magnification power. But the ocular lens is one only, fixed in a tube, and this part is called the nose piece, and this part is called the stage. This part is a mirror which reflects the light on the stage, and this is the adjustment, the fine adjustment and the major adjustment. And this is the part you hold the microscope from. It is called the arm. So, and this is the base of the microscope. There is many parts, some mechanical parts like the arm and the fine adjust and the large adjust and the base. These are called mechanical parts. And there is light parts like the ocular lens, the objective lens, and the mirror. This is the light parts. Okay. This you can buy one from the internet. You can search on the internet and buy one at your home. You can study some living organisms. You can take a drop of water to study. You can take a drop of blood to study, and so on at home. But after the year of 1950, the scientists start to make the electronic microscope. The electronic microscope. The electronic microscope the manufacture of electronic microscope start in the year 1950. The idea of the work is different from the idea of the light microscope. Why? Because they choose to make, instead of the light, they use a beam of electrons with a great speed instead of light. The beam of electrons has smaller wavelengths. They have smaller wavelengths. If you see here, the normal light wavelengths and the electrons wavelength. If we compare between the normal light wavelength, for example, like this, the wavelengths of the normal light like this, for example, wavelengths, you know, the distance between two tops or two crests or two troughs. This distance is called wavelength. You know this from your previous study. Type. Let's watch this is the wavelengths of the normal light, but the wavelengths of the beam of electrons is very small wavelength, like this. As you see, this is the wavelengths of the electrons. So it is very small wavelengths. So you can see more details. You can see more details if you have a smaller wavelengths. Here we have the smaller wavelengths. You can see more details of the sample which you study. This is a difference between the light microscope, which use the light, which has a large wavelengths, a long wavelengths, and the electronic microscope, which we use the electrons, which have a smaller wavelengths, a beam of electrons which have a smaller wavelength. And these electrons are controlled by electronic lenses, not glass lenses. The light microscope use glass lenses, but the electronic microscope use electronic lenses or electromagnetic lenses. Electromagnetic lenses. And this microscope has magnification power can reach 1 million times. 1 million times, and the photo is clear, still clear. But in the light microscope, if you make magnification more than 1500, the photo becomes unclear. But here in the electronic microscope, you can magnify the sample more than 1 million times. The importance of the electronic microscope, it helps the scientist to discover unknown cellular organelles that are more accurate details about the unknown structure. So now we can use the electronic microscope to see more details, more structures in the cell, and the fine structure of the cell. Electronic microscope is better than the light microscope because it gives a clearer magnification or magnified photos with high differentiation with high pixels. Show the wavelength of the electronic beam. The wavelength is very small, so you can see more details. It receives the magnified photo of the bodies on a fluorescent screen or in a very sensitive photo screen. You can receive the photo on a fluorescent screen or a sensitive photo screen. So the electronic microscope is better than the light microscope. You can magnify more than 1 million times, but the light microscope you can magnify only up to 1,000 times. And we have two types of the electronic microscope. One is called transmission and one is called scanning. The scanning microscope can give a photo for the surface of the cell. The transmission microscope can give a photo for the internal details of the cell. So it gives more details with more magnification power. So the transmission is stronger than the scan. The scanning microscope used to study the cell surface, only the cell from outside. The transmission microscope used to study the internal structure of the cell, more details. Magnification power can be 3,500x. Magnification power can reach 8,900x. So the transmission is stronger. We have some questions about what we have studied now. Are you ready to solve with me? Okay, let's start.
Yes.
Ruqayya, read the first one. Scientists give
the name cells to cork vacuum.
Yes. أول واحد.
Robert
Hooke أول واحد خالص.
Oh.
Okay. Second one.
Scientists invented a microscope that has magnification power of 200 times.
Yes. The second one. Who study the pond water and see the microorganisms in the pond water.
Hooke.
Very good. Hooke. Very good. Next.
Scientists stated that cells are produced from pre-existing ones.
Yes, the founder of the cell theory. Very true. Okay. Complete. Here we have the name. It doesn't give the name of the cell. We said before, Robert Hooke. State the animal cell, Van Leeuwenhoek. Number two, the light microscope is composed of two lenses, which are the two lenses in the light microscope. You remember? The ocular lens.
Light microscope.
Yes, two lenses. It has two lenses. Ocular lens and
objective lens.
Very good. Ocular lens
and objective lens.
Yeah. طيب, the second question. 3. Light microscope has magnification power of
1,500.
1,500. Very good. And
1 million.
the electronic microscope can reach of a 1 million.
1 million.
Okay. Next, scientists stated that the cell is the functional unit of the living organisms, while scientists state that all plants are composed of cells. نرجع كده نبص عليها بسرعة.
Cell is a function unit هيبقى روبرت.
شوف كده. الفانكشن يونت اهو رودولف فيرشو. Okay. The cell is a functional unit said by Rudolf Virchow, the function unit. Okay. The other one, he said that all animals are made of cells. Which one?
Either. Which one?
شوف كده. All animals are made of cells.
Theodore.
Van. Very good. Theodore Van. Very good. In the year of 1839. Okay. Next question. Write the scientific term, the building unit of all living organisms.
Cell.
A microscope which depends on the light and has magnification power 1,500 times.
Light microscope.
Microscope which gives magnification power 1 million times. Electronic microscope. Okay, I have only this problem. Calculate the magnification power of a light microscope, which magnification power of the eyepiece equal 50 and the magnification power of the objective lens 20. So the magnification power
1,000
will be the multiplication of 50 multiply 20. It will be 1,000 times. Very good. This is correct. And we come to the end of our session today. Inshallah, next time we start the fine structure of the cell. Inshallah. Thank you, Ruqayya. And I hope you study this part of the this chapter very well. I hope it's so easy for you, inshallah. Thank you and goodbye. Assalamu alaikum.